We were established in 1990, and we are a diverse group of recreationalist property owners, public agencies, and other folks. We provide annual student scholarships and host these annual meetings, and as well as have workshops. And if you aren't already a member, for those folks just showing up now for the presentations, please consider joining OLA. We want to make sure to thank our sponsors for sponsoring OLA and helping make this session possible, including CD3, IXOM, Biosafe, Phycotech, and Eureka Water Probes. Thank you so much. And then here's our session outline for today. So we'll start with Theo Dreher's presentation on Dolichospermum, and then we'll transition to some additional talks for the rest of the afternoon. I'll introduce those briefly at the start of their talks. So just want to quickly ask that folks keep yourselves muted unless you have a question at the end. And if you do have a question, you have two options. You can raise your hand, and that's in the reactions tab, or and I'll call on you at the end after the talk is finished, or you can type your questions in the chat box. And each speaker will have 15 minutes for presentation and 5 for Q&A, and then I will, for the speakers, I'll alert you at the 12-minute mark and at the 15-minute mark. All of these talks will be recorded and posted to the OLA website, so you'll be able to see them later. And I just want to remind folks, we do have one more session in this 3-part series. Our Beautiful Lakes, Past and Present, will happen on Wednesday, December 1st at 3 p.m. So with that quick introduction, I see folks still coming in, but I think we're on time, Theo, for you to get started right at 3 o'clock.
Okay, thanks, Desiree. Let's see, I just have to get out of there, get back to Zoom, share my screen. Oh, wrong one, dang. Okay, can you all see this screen, this introduction slide? Is that okay?
That's good.
Desiree, good, yeah. Thanks, Desiree. So I'd like to talk about the work we've done recently on Dolichospermum, or what we used to call Anabina. These are planktonic cyanobacteria that cause blooms in Oregon lakes, and some are toxic and some are not. And up front, I'd like to thank the co-authors, particularly Amanda Foss from Groundwater Laboratories. They've done the high resolution toxin analysis, and my colleagues Ryan Muller and Ed Davis at OSU, who've helped me with the genome analysis, and we've melded those together in this study. So over the last 10 years or so, we've looked at blooms that are caused by not by Microcystis, but by the Nostocales, the other common cyanobacterium that causes blooms, from lakes across Washington and Oregon, and particularly Oregon. And we focused on blooms that are caused by a single, or on samples where there's a single cyanobacterium present, because it simplifies the analysis. And so we've got a couple of lakes in Washington. Anderson Lake up there on the top left is significant because there we showed the presence of an anatoxin A producing Dolichospermum, which I won't talk about further. But you can see that in Oregon, there are a number of lakes that we've sampled. And that's also shown here. You can see the lakes down there in this lane. And so we've included AFA, or Aphanizomenon floss aquae, which has this typical morphology, and a bunch of anabinas, or Dolichospermums, as we are calling them now, and even a little bit of Glowiotrichia. And so the Aphanizomenons, floss aquae samples that we've analysed have not been toxic. That's also true of Glowiotrichia. But amongst the Dolichospermums, there are a number of non-toxic but also toxic examples. And those are the ones that I'll focus on. So this map here in Oregon shows where the non-toxics are from, these black ones. So they're from a number of lakes. Really, there are more non-toxic isolates than toxic strains that we found. But we have found cyanotoxin producers in a number of lakes, so that they're not at all uncommon amongst these Dolichospermum type cyanobacteria. So in Detroit, oops, sorry, I'm not doing it, so I'm just trying to use the pointer. In Detroit Reservoir, which I better not use the pointer, but we've seen the presence of a Dolichospermum, det 69, that produces Cylindrospermopsin, as well as the presence of a different Dolichospermum that produces microcystin. And we've also seen microcystin producers in Lake Billy Chinook across the ridge on the east side, further south in Lake Odell, near Willamette Pass, and in Juniper's Reservoir way down in southeastern Oregon. So there are actually a number of cyanotoxin producing Dolichospermum present in Oregon. Now, we've looked, whereas most analyses utilize ELISA, enzyme-linked immunosorbent assays, that don't give you any congener specific information, we're interested to find out which specific congeners were present, because especially quite recent studies, such as this one indicated under the microcystin section here by Chernoff et al., looked at oral toxicity in mice for different microcystin congeners. And so you can see there the listing of the MCLA through MCRR, and these are congeners or variants that have different amino acids inserted at this 2 and 4 position. And you can see that the toxicity ranges quite widely. This NOAL is the level at which higher doses lead to lead to symptoms and toxicity. And so LA, for MCLA, that boundary towards toxicity occurs at quite low concentration, so it's very toxic. Whereas MCRR is considerably less toxic in these oral assays. So we do want to know what congeners are present, because there's obviously a large impact on public health. And it's rather similar when you look at Cylindrospermopsins, where there are far fewer variants, but at this R position here, the 7 position, there can be 2 different isomers called syn or 7-epicyn. And there can also be a precursor, in which case there's still there's a deoxy position there in that position. And whereas the syn and 7-epicyn are quite toxic, the current information is that 7-deoxysyn is not toxic. So again, we want to know exactly what congener is present. And so what did we find? If we look at Lake Billy Chinook, where at the Perry South campground on the Metolius arm, there have been very high levels of microcystin recorded by samples taken from the Forest Service. And we found that Dolichospermum, which we named, oh crikey, sorry, again, oops, sorry, I've got this panel I can't get rid of, it's because I'm the host of this. I've got my picture up here, superimposing part of the slide. But Dolichospermum LBC05A was genetically found, that was the genome that we found in samples from 2 years from that lake, and identified MCLR, so one of the more toxic congeners, as the primary microcystin type in that lake. If we go down to Odell Lake a little further south, we have really the same result. This is again a lake that is subject to almost every season quite high levels of microcystin. And again, MCLR is the predominant congener. And there's a second congener with an ASP, that's a absence of a methyl position at that site, is also present at low level. But so in those 2 lakes, we have a pretty toxic congener as the main microcystin produced. And that's true also in Juniper's Reservoir, which we studied because in the summer of 2017, there were very high levels, as shown there, 3,000 nanogram per mil in this small lake on a private ranch, which cattle were drinking, and that led to the death of 32 steers. And in that case, again, MCLR with a little bit of the ASP3 variant is the main congener. So in those 3 lakes, we have predominantly MCLR. We didn't have the best samples for analysing the microcystin congeners in Detroit Lake, but in samples where we did have a detection, we found a quite different congener, and that's this MCH, which was the most predominant type, although it didn't account for all of the microcysteines that we think are present, because the samples that we had in this case had pretty low concentration. Although Detroit Reservoir can have elevated levels, as shown, and of course microcystin did break through the purification scheme at the water treatment plant for the City of Salem in the summer of 2018, resulting in low levels of microcystin in the water. And we found that a different Dolichospermum that we've called Det73 is the producer of this microcystin congener. So the congener is shown here, where at this 2 position, instead of the different amino acids, like particularly the leucine that we saw in the other cases, there's a homotyrosine. And at this 2 position at the top, where normally there's a methyl group added to, and the presence of this MDHA grouping, DHA, the demethylated form, was present. And that correlates very, very, very nicely with the gene sequences that we found for Det73, for these MCY genes, the microcystin producing genes, where normally there's this methyltransferase domain that is responsible for producing that MDHA. And this was just deleted. This whole small segment, that domain, was missing in the sequence of the MCYA gene from Det73. So that correlates with the observation of the demethylated DHA. So we've talked about 4 lakes. Turned out that in 2 lakes, Lake Billy Chinook and Odell Lake, we have really the identical strain present. And that's shown here, where we align the more than 5 million units of nucleotides of the genome, and see that where there are dark red ticks, there are very few of them. So there are very, very minor differences, and these are essentially the same strain. But the other 2 that I mentioned are very distinct. So we have really 3 very distinct microcystin producing Dolichospermums in Oregon. If we turn to Cylindrospermopsin, that was found in Detroit Reservoir. And there we have some really, really good samples with high levels of Cylindrospermopsin from 2015 through 2018. And in each of those cases, of the 3 variants that people have observed of Cylindrospermopsin, we saw 2. Predominantly the 7-epicin, so that's the toxic variant, and a small amount of the lesser toxicity 7-deoxy. And this typically has appeared in Detroit Reservoir at around Memorial Day at the end of May. So in Detroit Reservoir, then, there are 2 toxin producers, the Det79 that I just talked about that produces Cylindrospermopsin, or the 7-epi form, and Dolichospermum Det73 that is a microcystin producer. And we actually isolated the genome of Det73 with that missing methyltransferase domain, for instance, from a June sample there in the middle of summer. And the analyses of the toxins were from this, I'm trying to use the pointer again, from the Memorial Day peak at the end of May, where you can see there's a peak there. This is the 2018 data. There's a peak of Cylindrospermopsin in late May that coincides, has mostly or often coincide with some microcystin, and you can see that in the orange dots. This is a exponential y-axis, so it's not linear. And how are we doing for time, Desiree?
12 minutes, Theo.
Okay. So the typical distribution of presence of toxin that's been seen in Detroit Reservoir is shown here, where you have that peak of cylindro I was talking about in Memorial Day, and then some level of persistence, but pretty low throughout the rest of the year. Microcystin can be present at Memorial Day, but is not as high typically as the cylindro, but it can reach higher levels during the summer. And so the question is, are these 2 Dolichospermums I've been talking about, the 69 and 73, are they the only 2? Or are there other producers? And obviously we haven't analysed enough samples, but we think that those 2 strains could explain the observations and might be the only 2 major producers. But it'll be interesting to look at that in more carefully. So just to summarise, we've actually seen really a large diversity of Dolichospermums in Oregon, both genetically and in terms of their toxigenicity. And if you, so we've got the non-toxic and the 2 toxin producers, and then if you include Washington, there's a 3rd major cyanotoxin producer. So it's a really important grouping that can be quite problematic in various ways. In looking at the microcystin congeners, we've seen that MCLR, which is pretty toxic, is seen in 3 lakes. And the demethylated hydrotyrosine variant, the DHA7 MCHTYR, is found in Detroit Reservoir. Now, I might say that we're not 100% super sure that this is the correct identification, but we're pretty certain of it. So certainly a quite different congener is present in Detroit Reservoir, and its toxicity hasn't been well studied, so we don't really know what the public health consequences are. In terms of cylindrospermopsin in Detroit Reservoir, that's made by Dolichospermum det69, and it's the 7-epimer or isomer of cylindro. And then finally, as I mentioned, we think there are probably the 2 toxin producers in Detroit Reservoir, although further research conceivably could find an additional one. So I'll just finish by thanking my colleagues and the university for funding. Thank you.
Wonderful. Thank you so much, Theo. We are virtually clapping. Do folks have any questions for Theo?
All right, I'll leave up this summary slide if there's any questions out there.
Well, Theo, one question I always have about these kinds of studies are how managers might use these results.
Yeah, you could certainly develop strain-specific probes, and if you know the range of players of bloom-forming cyanos in your lake. If you study it for enough years, and we've done this in a few lakes, we find that it's actually pretty consistent. And so you could even have morphological observation to be to sort of have a warning, have an indication of what the probable expectation in terms of toxicity is. So I think it's about knowing what the products are and which the cyanobacterial players in a lake are, what their potential is, figuring out which ones are the ones you're concerned with, and that would, often be the cyanotoxin producer, and then finding ways to track that. And you can do that genetically. Clearly, that we could develop probes for that, and there are other ways too that one could use once one has really described the system. And I think it's important to know which strain that you might see in a microscope is the producer of which cyanotoxin. Get those 2 things together.
Okay, great, thank you. So we've got
Mark, you've got a question?
Mark, and then Laura, and then Amalia, and Tamia.
All right, thanks. Thanks, Theo. That was very, very interesting. Are you finished with this? Are you anticipate doing any more work on this? Because the lake I'm working up on in Washington is Dolichospermum blooms every year, pretty toxic, and if you're interested in more samples, I could help you out there.
Yeah, since I retired, I don't want to start too much, but I sometimes can't resist something that's really interesting. So maybe talk to me.
Okay.
Laura,
you had a question?
Yeah, Theo, I was kind of curious, you brought up Diamond Lake not having a toxigenic strand, but it wasn't clear, I may have missed it, is how many samples you got from that lake?
No, these are not comprehensive analyses of these lakes. These are sort of samples taken when we knew about a bloom and could get a sample suitable for genome sequencing, which, as I said, is when there's one predominant strain. So all we can say is that there was a Dolichospermum in Diamond Lake that was. Doesn't say that there not be different ones at different times at all.
Yeah, I guess that to finish that thought, do you know offhand how many samples would be required to be comprehensive to get an idea of a lake's like strands, or is it?
Well, we've followed Dexter Reservoir over a number of years, so that we think we've got a really good picture of it. You just follow it all the time. You figure out we went for weekly sampling. You figure out what at any given time is a significant part of what's blooming, and try to identify those. And one could identify those in terms of genetic sequences. That would be the best way. You would want to identify them microscopically because sometimes they can be distinctive, not always, but sometimes it can. And then you can, when we've done that in Northwest Lakes, we find that they're mostly fairly simple. There aren't that many different players. Now, it might change at one given time, but we've looked at a number of lakes over a number of years, and they're really pretty stable. So I think that's what you'd want to do, weekly sampling over a few years, I think, just to have an idea of what's present.
Cool, thank you.
Jamila, I think we're going to have to move. Oh, go ahead.
Can I ask a question now?
Sure, go ahead.
Yes, just a short question, Jamila.
Sure. So just, I'm wondering if you have research conducted at Gold Lake, because there is this report which was done in 1982 talking about presence of 2 species of cyanobacteria. I'm asking this question because during the isotopic analysis of the Gold Lake, the isotopic nitrogen is very negative, like at the Gold Lake. So I was wondering if you have worked at that lake recently, or is there any planning to work on that lake? So that will be really helpful, what we have found through the isotopic analysis.
Which lake is that?
It's Gold Lake in Willamette National Forest, on which I'm working.
No, okay. I haven't. Sorry.
Thank you.
Yep. Thanks, Desiree.
Thanks for all those questions. It's a great discussion. Our next speaker is Emi Fergus with the EPA. Take it away, Emi. Thanks.
Hi. One moment. Let me get this set up. Can you see my slides okay?
Yep, it looks great.
Okay, great. So yeah, thank you. It's been really fun listening to the talks from last week and Theo's talk just now, just to get a more inside look at Oregon lakes. I'm going to be taking us out to a broader landscape range and present work that I've done with my co-authors listed here as a postdoc with the USEPA based in Corvallis. And we've looked at developing a lake hydrology model to understand the variation that we see in hydrologic characteristics in US natural lakes and reservoirs across the country. So just to give a little background information, lake water levels are a defining characteristic of lake ecosystems that affect ecosystem function and the services that humans enjoy. They affect things such as the nearshore habitat condition, what type of biological community is present in a lake, as well as things like biogeochemical processing and nutrient cycling. And lake water levels naturally fluctuate just due to natural imbalances in water inflows and outflows related to climate and watershed hydrology. But human activities on the landscape and dams can alter water level fluctuations beyond natural ranges and impair lake ecosystems. On top of this, changing climate may exacerbate these effects. And because of this, altered lake hydrology may be a widespread and serious threat to lake condition, but we don't have a great understanding of the natural and human drivers of these changes, especially when we're looking across different regional settings. And so for this project, our objective was to quantify the relative influence of natural and human drivers of lake hydrology in lakes and reservoirs across the United States. And to do this, we first developed a conceptual framework of hypothesized drivers that affect lake hydrology, and then translated that conceptual framework into a path analysis model using structural equation modeling techniques to test that representation. And we applied this at a national extent as well as across 5 ecoregions. And for this talk, I'm going to focus just on results from the West and Midwest to just show contrasting differences in different drivers of lake hydrology. So our data come from the National Lake Assessment Surveys, the 2007 and 12 data. And if you're not familiar with it, it's a rich data resource that sample a wide range of lakes that are representative of what you would expect to see on the landscape, following a probabilistic design, sampling design. Our geospatial information comes from the LakeCat geospatial database and includes things like climate, land use, watershed hydrology, and other things. And then we used the Hydra Metric as our representation of the potential for humans to alter lake hydrology. And I'll talk more about this a little bit later. So the lake hydrologic variables that the NLA records include lake water level drawdown or decline, which you can think of as like bathtub rings. So the NLA field crews go out and they measure the decline in water levels from the historic high water lines around the lake. We also have this parameter called evaporation-to-inflow, which is derived from water stable isotopes. And it represents the proportion of inflowing water that is evaporated, such that values close to 1 indicate more evaporated water in a lake. To try to tease apart the natural and human drivers of lake hydrology, we needed to have a measure of human disturbances that can impact lake hydrology and that could be applied at a national extent to the diverse lake types that we have in the NLA, which may not have a lot of monitoring presence. So it has to rely on geospatial information. So we developed this thing called the Hydra Metric, which is a national scale framework that ranks lakes by the potential for human hydro alteration based on dam attributes and land use activities that can alter watershed hydrology. And we ranked the NLA lakes on a scale from 0 to 7, where 0 indicates no apparent human hydro alteration and a 7 indicates high potential for human hydro alteration, as indicated by large capacity dams or land use activities like irrigated agriculture. And from this map, you can see that in our 2 ecoregions that we're going to focus on, there's quite an array of human hydro alteration potential on these lakes, color-coded there. So just to give a sense of the variation in lake hydrology that we're trying to model in the US, here we've plotted box plots of the distributions of our 3 hydrologic response characteristics that we have: evaporation to inflow, as well as water level decline. And we've plotted it against 5 ecoregions delineating the continental US. And for this talk, I've pulled out Oregon lakes in the NLA just so you can get a sense of where Oregon lakes lie across this distribution. And just eyeballing it, you can see that there are definite ecoregional patterns and variation in these characteristics. Western lakes tend to have low E to I, indicating that they're more flow-through systems, and they tend to have greater vertical and horizontal decline, whereas lakes from the Midwest span a broader range in E to I measures, ranging from, from more flow-through systems to more closed basins that experience more evaporation with these higher E to I values. And they tend to have smaller vertical and horizontal decline. And so we wanted to see if our model could kind of account for some of these patterns that we're seeing. So we developed a conceptual framework based on literature and theory of hypothesized natural and human drivers of lake hydrology. And they included things like lake morphometry, land cover, hydrology, and climate variables, as well as dams and land use. And we hypothesized that these natural drivers would have pretty robust and consistent effects across ecoregions, but that we might see regional variation in how human effects alter lake hydrology or influence lake hydrology based on different regional climate and how humans use water across ecoregions. So we took that conceptual representation and translated it into a path analysis model using what data we had available with the NLA and LakeCat data sets to predict our three response variables, E to I and water level decline. And we used various predictor variables that we can group by driver class that include things like watershed hydrology, climate, lake morphology, as well as anthropogenic factors, which we are representing by that Hydra metric. We also were interested in looking at whether there was evidence for an interaction between drought, represented by the Palmer Hydrologic Drought Index, and our Hydra metric, whether that might influence how lake hydrology reacts. So what we found was that our path analysis model fit the data pretty well given SEM modeling performance criteria, and that the model explains similar levels of variation across our three response variables in both the West and the Midwest. So it explained over a third of variation in E to I, between 21 to 38% of variation in vertical decline, and over half of the variation of horizontal decline that we saw in these ecoregions. And below are the path analysis models that you can see that the black arrows represent positive relationships and the gray dashed arrows indicate negative relationships. And the thickness of that arrow indicates the magnitude of those relationships. So although the model accounted for similar levels of variation, the drivers and their pathways were pretty different between the two ecoregions. And this is only showing part of the picture. These path models are showing the direct effects, but one of the really cool things about SEM is that you can also get at the indirect effects of how other variables may be moderating the effects of others. So to really understand how these predictors affect lake hydrology, we should be looking at their total effects, which I'll show next. So here I've plotted the standardized total effects of our individual predictor variables that are color-coded by driver class for each of our hydrologic response. Oh, I'm sorry, got a sensitive mouse. Against our three response variables for the West and the Midwest. And the plots show the direction and the magnitude of those effects with the horizontal line being 0. So you can get a sense of the direction of those relationships. So just looking at E to I, we found that lake depth and watershed inflow had a negative relationship with E to I, indicating that shallow lakes with less surface inflow tended to be more evaporated lake systems or have higher E to I. We also found that in the West, that Hydra Metric was associated with lower E to I values, indicating that these systems tend to be more flow-through systems, which you might expect being familiar with the region and what types of lakes are present. Whereas in the Midwest, that Hydra metric didn't have any significant relationship with E to I. Looking at vertical decline, we found that lake area and drought were associated with greater water level declines, but the magnitude of drought effects were much more pronounced in the Midwest than in the West. We also found that Hydra metric was related to vertical decline in both the West and Midwest, but they had opposite effects. In the west, hydrap had a positive association with vertical decline, whereas in the Midwest, it had a negative association. And this implies that where there is water management or water infrastructure present, in the West, it tends to lead to greater vertical declines, whereas in the Midwest, it minimizes decline and maybe leads to more stable full lake levels. We also found evidence for an interaction between drought and hydrap in the Midwest, which was interesting. It shows that when lakes are in drought conditions, hydrap or having dams present in the Midwest may minimize the effect of drought on vertical decline, such that it kind of dampens the effect and you have more full lake levels. And so what we took away from this is that this might be capturing how lakes are managed differently across these different ecoregional settings. And finally, looking at horizontal decline or littoral exposure, we found our model did a good job of explaining this, and this was largely because we included vertical decline as a predictor. And that had a very strong direct effect. We also found that lake basin morphometry influenced horizontal decline, such that shallow lakes with gently sloping banks had greater littoral exposure. We also found that climate variables affected horizontal decline, but indirectly through their effects on vertical decline. So just some takeaways from this. Our path analysis model showed that the factors related to lake hydrology are different across ecoregions. In the West, water level decline is largely related to human hydro alteration potential, where dams and other human disturbances are associated with greater water level decline. In the Midwest, it was more largely related to climatic factors like drought, and where there was human hydro alteration, it tended to promote more stable water levels. So it's going to be important to try to untangle how natural and human drivers affect lake hydrology, especially during changing environmental conditions, to support lake management that can protect lake hydrologic integrity. And our intent was that this work would provide a landscape level framework to evaluate multiple drivers of lake hydrologic variation that can be adaptable as new information and data become available. And with that, I thank you for your time and can take questions if there is time.
Thank you so much, Emi. Yes, yes, we do have time for questions.
All right.
Looks like we have one from Tamara Wood.
Am I on?
Yes.
Yeah, okay. So I was having a hard time following, well, not following, but it went by pretty fast, but it looked to me like temperature in particular had a different direction of influence in the West and Midwest, and that may have been true of a couple of other things too. And I was trying to sort of wrap my head around that. Do you have an explanation that you've worked out for why the temperature influence in particular would have a different direction?
Yeah, I don't know if you can see my screen now.
I can see your screen.
Okay. Yeah. So for water level decline, so the measures we had were annual summaries of mean annual temperature, which may not be the best temporal scale to be getting at lake hydrology that's been measured during those summer visits when the NLA crew are out there. So that's one thing to consider, like maybe the annual scale is not appropriate in capturing what's happening, how temperature is affecting lake hydrology. And so it is hard to see because it's so tiny on here, but I think we were finding that warmer temperatures were associated with greater vertical decline in the Midwest. In the West, it really didn't have a strong relationship. And again, that might be because we should be looking at a seasonal scale and not an annual scale. And those are things that, yeah, we'll need to tweak and play around with more. But as a first pass, we thought we'll just start with the annual, see how that goes.
Thanks.
But yeah, thank you.
Amalia?
Hi, Amy. Thanks so much for this talk. It was really, really interesting. I'm wondering if you could talk a little bit about what was involved in the lake morphometry piece of the models. And specifically, I'm kind of interested if you've looked at kind of depth relative to the perimeter or surface area of the lake, or whether that's kind of embedded in this evaporation inflow metric.
Mm-hmm. Yeah, those are good questions. We kind of just took the simpler morphometry measures that we had available in the NLA, like maximum lake depth, and NLA field crews went around and kind of gauged the littoral or the bank slope and had an index value for that. So we were using their observations to get at that bank flatness measure was what we had in this model. I thought about playing around with like lake area and depth or some other things, but we didn't get that far with it. I think seeing how much lake morphometry plays a role in understanding E to I and littoral exposure especially, it's worth trying to explore different morphometry metrics, which we haven't done for this stage yet, but something definitely to consider. So it's a good point.
Thanks.
All right, maybe one more question from Jamila.
Hi, Emi. Thank you very much for the great talk. My question will be specifically to the wide range of temperature over the lakes that you have studied. So I was wondering, did you consider studying the particular lake for the temperature variations over the period of time? Because there are multiple studies in the part of the US where the chironomid fossils have been used to infer the temperature over the period of, for the last 15,000 till now. So I'm just suggesting like it will be a great idea to just aid that thing to see how the temperature has been fluctuating over the period of time into these lakes that you have been studying.
Yeah, I think those are really great points. We kind of looked just at that annual scale, but it's not probably showing the full picture, especially that E to I measure. It's water stable isotopes that were collected at the time that the crew were out there. So having temperatures more closely aligned to when that was sampled would probably help give us a better picture of how these drivers are affecting the characteristics that we have. And yeah, I think it would be interesting to look at different timescales for temperature and other climate variables and how we can find some kind of alignment with the variation we're seeing in hydrologic characteristics. So thank you. Yeah, that's a good point.
Yeah, specifically for those lakes where you have seen like a dramatic change in the hydrology of the lakes. So studying those lakes in detail using those climate proxies and projecting in the future how those will be behaving to the temperature changing with the climate change. That will be a great addition to the current studies. Thank you.
Yeah, thank you. It's great.
All right, thank you so much, Emi. We are going to pivot now, and y'all are stuck with a presentation from me. I'm going to give the next talk, and we're going to pivot to a conversation around harmful algal blooms and using the use of models to examine options for controlling HABs. I'm going to use my laser pointer here and just going to quickly introduce this concept of what's essentially the Ross Island Lagoon and what's happened out there historically. We can see on the left figure here is the historical context for the lagoon, where you had two islands here, Hardtack and Ross Island, and they were connected by the core earlier in the century. And then over the whole last century, there's been a lot of gravel extraction. So now there's a large stagnant pool of water in the middle of the river. Its deepest point's right around here. It's around 120 to 130 feet deep. And it warms up in the summer, produces harmful algal blooms. And in years that are particularly severe, you get water that the tide comes in on a like base flow. There's maybe 3 foot of tidal excursion in the lagoon. And so as the tide leaves the lagoon, it drags some of that algae downstream. And as we saw in this example from this year, there was a pretty severe bloom, and OHA advisories were issued as a result for the access to the river. So it's a high profile project. We've been working on it now since 2017. And so what I'm going to be presenting today is just a quick overview of some modeling work that we've been doing. And this is just preliminary model work, and with the key goals of trying to understand what's going on out there currently, and then to do sort of a screening level analysis of some different alternatives. So how can we potentially address this issue? There are obviously chemical treatments that can be applied, but because it's connected to the river and directly connected to the river, and there are threatened and endangered species in the system, a lot of the options are not on the table. So we've been trying to look at mechanical or physical processes. So one of them is, just to briefly give you a summary, there's this sort of spit of land right here. And the idea is to remove this, to essentially excavate this land out, and that would increase the tidal influence. And this is the Holgate Channel here. The river is flowing in this direction, and it splits around the island now and goes in this direction. So if you increase the influence of the tide, could that help mix the lagoon and suppress the stratification? A second is just considering aeration of the bottom of the lagoon. A third one, which I'm going to focus the most on today, is mechanical pumping. As you'll see, that the bottom of the lagoon is really, really cold. And so if we bring some of that cold water up to the surface, will that help suppress productivity? And then the last one, there's a lot of attention and interest in, is the idea, essentially, this is where that berm was constructed. Could we essentially cut a hole in it and let the river flow through here again as it used to? And it is a particularly complicated option because there's some contaminated sediments buried in this lagoon, in this part of the lagoon, some pretty nasty stuff, and like tributyl-10 and other things. So that's a particular issue as well. There's some shallow water habitat that's been constructed here that people are particularly attached to. So it's not an easy solution, but over the long term, it doesn't involve burning energy to solve the problem. It essentially reverses the issue that created the stagnant pool in the first place. So I'm going to give you a very, very quick overview. Scott's going to talk about modeling next, but we used his W2 model. And it's WC equal W2 is a laterally averaged, well, we applied it as a laterally averaged model. So it represents the channel at depth, and which is an important feature of modern lakes, obviously. So we have one straight cross section that cuts through the deepest point of the lagoon, and then we have a bunch of horizontal segments that cut across it. And you can see, actually, this is a LiDAR plus a bathymetry site. It's a bunch of different topographic data, and you can see in the bathymetry this deep hole that's right here, and then how it gets more shallow in some of these other areas of the lagoon and the river over to the left. So this just gives you a sense of that cross section. This is essentially what the bottom of that cross section looks like, and that for these model simulations, we were calculating, making all the calculations of water, the hydrodynamics, the water quality, the algal growth at 1 meter vertical grid spacing. So we had 40 total layers and 20 of those segments across the lagoon. So I said it was going to be really brief because I've got 15 minutes to tell you a lot about what the results are. And one of the key ones that I think is really important here is that groundwater appears to be really contributing some cold water and some nitrogen at the bottom of the lagoon. So and I'll explain why this is in quotation marks in just a second. So I'm going to show you both some model results and some profile data. So I was out there in 2020 collecting a bunch of data every 2 weeks. And these just show you, the orange show you some profiles that I measured. with depth. So this is in April and July, and then during more of the bloom season. And what you'll notice is that as it gets really cold, it's 8 degrees Celsius at the bottom of the lagoon consistently throughout the summer. It was between 8.1 and 8.4 degrees Celsius. And so I should have mentioned Kelly Vacher is the person who's done the modeling for this project, and in collaboration with myself and some of the other project partners. And let's go ahead and get the model started. As we started to simulate the system, we noticed that if you don't represent groundwater in any way, which is what's here on the bottom, as the system starts to heat up, as the surface starts to heat, the entire lagoon starts to warm up. And that's just not what we saw. Essentially, like standard lake limnology or lake dynamics don't explain this pattern that we saw with it getting really cold at the bottom. So Kelly added a continuous injection, like discharge and recharge and discharge of groundwater into the lagoon. So it's coming in, it's leaving. And that's what it essentially required for us to reproduce this cold pool at the bottom of the lagoon. And so you sort of see that here, that's the blue line where we have groundwater represented up here in the upper left versus if no groundwater is represented. And it's not a perfect fit. We have 1 year data. And this is really sort of a preliminary model, but it was really illustrating the point that there's this exceptionally cold pool of water at the bottom of the lagoon. And on the one hand, that's a bad thing because it means it's contributing to really strong stratification, particularly in the summer. On the other hand, that cold water could be a resource in terms of helping suppress the HAB if we bring it up to the surface. So one of the questions that's coming up, though, as we think about, pumping water to the surface is, what is the source of that cold water? So this is just these are the temperature profiles that I measured throughout the year. You can see at the hypolemnion, it's just really cold and stays cold. One of the key things, though, about calling it groundwater is that the regional groundwater is discharged at around 12 to 13 Celsius. So the pool at the bottom of the lagoon is consistently quite a bit colder than that. And so the question is, where is that cold water coming from? It can't be groundwater because it's too cold. And so I've been spending a lot of time thinking and talking to folks about groundwater dynamics in the lower Willamette. And the best sort of storyline that we have is that that's coming from the river. So if you go to the USGS record, discharge in January and February is on the order of like 6 degrees Celsius. So it's possible that this is actually river flows that are being discharged, stored as like hyporheic flows and being discharged into the lagoon in the summer. We don't have confirmation of that, but that's sort of our current working hypothesis with this preliminary model. Very much open to other suggestions or alternative ideas about what might be going on, because this is really still in the phase of trying to figure it out. But we are hoping to kind of help understand that better, because the concern here is that if you start drawing up that cold water, that we're essentially going to create conditions where the lagoon could turn over and we essentially lose access. We lose that cold water pool. And so we need more modeling to understand what pumping rate would actually do that. It's a big water body, there's a large volume in there. And we also are looking to conduct some isotopic tracers over time. So to figure out not even the volume, but just where is that water coming from, to confirm that this is actually river water that's being stored. So we would be collecting isotopic tracers from the river, from the hypolemnion of the lagoon, and from a nearby groundwater well over time. And so to see if the signals start to converge between winter river and the summer base hypolemnion. So still sort of an open question, but the models at least help us understand there's something interesting and important going on here. That's the sort of current conditions. And then going forward, I'm going to be very brief about some of these alternatives. But expanding the entrance, as I suggested, sort of opening up, essentially isn't going to do anything isn't likely to be effective in terms of increasing mixing. And so this is an illustration just for the hottest part of the summer. As the tide comes in and goes out, you can sort of see this internal seiche that it sets up and kind of at least creates some small disturbance in the metalemnion, but does nothing to actually break up stratification. So even expanding it really wide, the tide's not likely to do what we need it to in terms of suppressing the algae. Hypolimnetic aeration is also not likely to be effective. A lot of data here. I just want to draw your attention to this idea where we injected air in the bottom of the lagoon, and it does increase dissolved oxygen concentrations, but it doesn't do anything to suppress, for example, release of phosphorus from the system, or over here with cyanobacterial growth, it doesn't really impact cyanobacteria concentrations either. So minimal impact. We can probably eliminate that as our solution. And then finally, our last option that we considered in detail was pumping. And this is, again, the idea that you bring some of that cold water pool up to the surface. And the key finding here is it's likely to be aggressive and it's not going to be cheap to pump that much water. And we could actually reduce the surface dissolved oxygen. So just to briefly show you what we did, this is that cross-section essentially of the lagoon that I showed you earlier. And we essentially sort of somewhat randomly, but picked a cold cell within the model and brought that water up to the surface. So moving from the yellow to the red box. And then we analyzed it in a number of places across the lagoon. In this case, the results I'll be presenting is just this green box near the surface. And then we did that at a bunch of different pumping rates. So starting at 250 gallons per minute up to 3,000 gallons per minute. So that's what these results are showing you. There are visualizations of the peak biomass for cyanobacteria. So this is an example in August 10th for no pumping, 250 gallons per minute. You see it's not changed dramatically. Starts to reduce biomass as you increase up to 1,000 gallons per minute, but really starting to see some benefits once you get up to 3,000 gallons per minute, which is a really high pumping rate. So it's moving a lot of water, but it's starting to be effective at reducing biomass. And essentially the mechanism for this is essentially that it's cooling the surface. So you see that this is, these are the pumping rates where this blue is your 3,000. So we're seeing the temperature drop, not by a lot, just by about a degree, but that's been effective at reducing cyanobacteria, which is illustrated here. We also modeled diatoms, which I kind of think of as like the good guys versus the bad guys. But notice, so they also dropped, but notice the range on the x-axis or y-axis here is, you know, an order of magnitude smaller than for cyanobacteria. So yes, the good guys sort of also dropped in terms of productivity, but because they're not as sensitive to temperature as cyanobacteria, we see a much bigger drop in cyanobacteria. But roughly these are expensive. They're around $200,000 per pump. A big chunk of that is the generator, which is going to burn a bunch of fossil fuels. So it's not an ideal scenario, likely to be effective to some degree over the short term. But then the question is, what does effective mean? Like, how many of these pumps do you actually need? And that becomes this whole other thing, which has now been published. There's a couple papers out in journal articles of like, what does it mean to control a HAB? What does it mean how do you even define what a HAB is? And so the lagoon is currently listed for chlorophyll A at a threshold of 15 micrograms per liter. But it's an imperfect measure for reasons probably most of us already know. One is that both diatoms and cyanobacteria produce chlorophyll A. And so in 2020, the year I was out there collecting these data, this 15 micrograms per liter was exceeded only for a few days, even though there was clearly a bloom going on. Nobody collected data in 2021, but I suspect that value was much higher. It was a much bigger bloom. So one sort of caveat or explanation or outcome of this is that we need to modify those pumping strategies from between years if we're trying to achieve this 15 micrograms per liter. And the other outcome is, well, so it's really easy for modeling the alternatives. It's not perfect because it lumps essentially everyone together, as I alluded to earlier. And that's what's illustrated here. These are just early dates in the 2020, and then these are later dates, August 5th versus August 27th. And what this shows is that these different pumping rates have very different effects. In early, as you often see in lakes, we have this bloom of diatoms. And pumping actually, if we go from the blue to orange, the first pumping rate actually increased production of diatoms, likely because it's bringing up some of the nitrogen that was at the bottom at the hypolimnion. But then when you get here, the pumping up that cold water clearly knocks back the cyanobacteria when they were that were associated with this peak in chlorophyll A. The other thing to point out, though, is that neither of these are anywhere near reaching the limit, the threshold for the TMDL. So the last finding was just that we didn't essentially get the flow through the cut in the channel model because it's a much more complicated model than what we were able to do with the resources we had. So I just wrapping up with these key findings. We know that the tide and aeration aren't likely to be effective. Pumping is, but it could be expensive, both from a cost perspective and a carbon perspective. And then we need to do more work to really understand the flow through channel. But even related to pumping, we got to figure out what the source of that cold pool is. Are we going to exhaust it by pumping 3,000 gallons per minute? We need, I think, broadly more conversation around what it means to control a HAB. I'm not convinced that 15 micrograms per liter of chlorophyll is the appropriate target, but it's the regulatory target. Yeah, and then we need more modeling, as we always say. So anyway, thanks so much for this whirlwind tour of the lagoon, and I'm happy to take questions. Thanks. All right, I see questions first from Laura and then from Scott. Laura.
Great talk, Desiree, and this is super interesting. I was kind of curious, maybe this is because I'm not familiar with this model, but just knowing as an ecologist that like cyanobacteria is pretty diverse, like what is the uncertainty with the cyanobacteria response? Like, is there any uncertainty measure with this model, like that you're incorporating? Because it can be variable.
Yeah, that is a great question, Laura. And I can tell you that there is definitely uncertainty associated with it. There's a bunch of ways, like the model sort of partitions chlorophyll A to represent who's producing it. There are ways that we've had to sort of parameterize the model to represent the cyanobacteria versus the diatoms. And all of those are, they're imperfect. And like I said, this was a shoestring budget that we got to do this preliminary modeling. So a sensitivity analysis, there's a bunch of things that we should be doing to have sort of final results, but the goal here was really that screening level, which analysis of which ones do we think are likely to be effective before we'd ever make a recommendation, for example, of using these pumps to bring the cold water up. We definitely need tighter control and better data and sensitivity analyses to really understand production, the cyanobacterial production, because we essentially just represent them as like parameters of how fast they can migrate in a day. So it's, yeah, we're certainly not representing different species. Like we know that Aphanizomenon's out there, Microcystis is out there. We're just lumping them all together as cyanobacteria. So it's not sensitive to that, especially this sort of screening level analysis. Great question. And Scott, I'm afraid to hear your question.
Don't be afraid. Thank you very much, Desiree. It was great to see the fruition of your work out there. I had two questions. One, and maybe it's not the right place to bring it up or talk about it, but the boundary condition between the little lagoon and the river, how was that set up? And then, because that's pretty critical in terms of getting the right temperature and water level variation in the channel and having it communicate with the lagoon. And second, I noticed that in your profiles of model predictions with and without groundwater, it was clear you had too much wind in your model. And that could be another factor why you're apparently thinking that the groundwater gets used up, because if there's too much wind mixing, it'll mix it all the way down to the bottom. So, and as you know, and I've kayaked in there millions of times, it's a lot calmer in that lagoon than out on the river. So anyway, just depends on the wind and what that is. That could be a big factor.
Okay, that's great feedback, Scott, on the second point. Although I will say it gets so windy in there. There were so many times where I've got an oar raft that I'm pushing and up into the wind, and there were literally multiple times where I thought, can I just drag this uphill just to avoid like paddling into the wind? It was but our data, yeah, I think our wind data came from the airport, so it is not a local. They're not local wind data. Because we didn't have it. So that's a really important point and something that we can definitely look into further with the next phase of the modeling. But regarding the boundary conditions, so yeah, it's a sort of convoluted answer, but there's the Morrison Street gauge that's downstream. And so we developed a 2D HEC-RAS model just to first to start to understand the hydraulics of the river and the lagoon and its connectivity before we did the W2 model. So we had the boundary conditions that were simulated using the Morrison Street Bridge data, the USGS gauge. So the boundary condition at the entrance to the Holgate channel is modeled as an outcome from that 2D HEC-RAS model.
And does that include temperature too?
Yes. Yeah.
Great. Thank you so much, Desiree.
Yeah, thank you. Great questions. Okay, so we're going to now transition. Oh, sorry, Theo, we're out of time. If you don't mind putting
Okay.
that window.
Yeah,
sorry
okay, go
to cut you
ahead.
off. Scott's our next presenter, and so I want to turn it over to him to talk about using these types of models and sort of best practices. Scott with Portland State, turn it over to you. Thanks.
So hopefully you can see the screen. Okay, is that all right? Okay. So I'm going to just talk about how good does your model have to be. This is kind of a little tongue-in-cheek sometimes, but anyway, what I'm going to try to talk about is basically, 1 minute. Is your model good enough? And just go over a few quick reminders, some steps in model development, some comments on model calibration and calibration guidelines is really my final thought as to how good does your model have to be, and a couple final thoughts. So also when you do modeling, remember that sometimes there are no bad models, just bad modelers. And I have to be part of that as well. I have been a bad modeler, and we shouldn't take it out on our computers. So anyway, it's not the computer's fault. We should be looking at the mirror sometimes, and I've been guilty of this many times. So one of the great advances in our technology is that we've got incredibly powerful computers that allow us to get the wrong answer faster. And so we have to be careful about how we manage that. And anybody can run a computer anymore, or a computer model. So we have to really be careful about the process and make sure that it's good enough to move forward. So typical steps in model development, typically people set up a model, they go through calibration, then they go through what's often called a validation step, and then application of the model. I'm gonna just say that I don't believe in model validation because I've never seen anybody do it correctly. And so in my mind, it's all model calibration. If you calibrate for 10 years, then and you run your model for 10 years, then you've calibrated over 10 years of time. And to actually get a statistically independent data set and to prove that it's truly a validation is a stretch. And so anyway, I'm not going to talk about that today, but that's just kind of my own personal idea. Or not my own, but others have that too. So because of that, this is kind of the revised setup that we usually go through, set up the model, calibrate it, and then in green, we always have to ask ourselves if we're calibrating for the right reasons. And that's a tough question. Sometimes it's pretty obvious. And then once you feel like you've done it for the right reasons, is it good enough to actually apply the model? So those are some of the processes that we go through. And really, when we look at the traditional view of model calibration, you have what you measure, and then you have the real system on the top and the modeled system down here, which is the computer model. For the real system, though, it has unmeasured inputs and measurement errors. And usually you go through an adjustment of model parameters until the error is acceptable, and then you're done. But that often isn't good enough, because usually a revised view of calibration should undertake to look at the field data and try to understand the unmeasured inputs to the real system. So a lot of our work is not twiddling with model coefficients, but really just re-examining inputs to the system. Do we have all the measurements? If we don't have them, how do we estimate the unmeasured inputs and adjust the boundary conditions? And then hope that without much model data comparison or model parameter adjustment, you can get close to getting an acceptable calibration. And in some cases, the data is wrong. So sometimes people have the idea that, field data is sacred, but sometimes the model is correct and data are wrong. I don't want to say that in general, but sometimes you have to look at that. And I'll give you an example. So what are some examples of calibrating for the right reasons? On the screen on the left is the Willamette Falls at Oregon City, and on the bottom is Bonneville Dam on the Columbia River. And in one model of the Willamette Harbor, people actually moved Bonneville Dam 40 miles upstream, and they moved the Oregon Falls 70 miles upstream the Willamette in order to match data of tidal exchange in the Willamette Harbor. Obviously, that is totally incorrect and is calibrating for the wrong reasons. Another example is, what if you have a model where your temperature in your model is too high and your data are much cooler? Well, one smart way to take care of that is to reduce the surface area in your model until you get the model to match the data. Again, calibrating for the wrong reasons. If your system is 10 meters wide, your model should be 10 meters wide. And I've seen examples where people have taken a model and moved it to 5 centimeters wide to cut out the energy, but it should be 10 meters. So obviously these are egregious examples of calibrating for the wrong reasons. And another aspect of that calibration has to do with quality of field data. Look at an example of the Willamette River Basin model. This is the Willamette River main stem all the way through the Newburgh Pool, the falls, and then into the Columbia River. And I asked Desiree about this because she could use this for the lower Willamette if she wanted to as boundary conditions to her lagoon system. So one of the issues was on the Mackenzie River, they had field data at these sites that are in green. And when we did model data comparisons at most of these sites, the red is the model and black is data, you know, we had incredibly great diurnal response of the model compared to field data, the Mackenzie River. Also, if we compare daily maximum temperatures, model versus data, it was quite good. But then if we looked at a summary of all the errors at those different river miles, you might notice the mean error, ME, that should be close to 0. AME is absolute mean error in Celsius, and RMS is root mean square error. And we're hoping that AME is all less than 1 degree C, and it is, except for this one data point at river mile 15.61. And we finished our report and said we couldn't get that data to match. And we're very fortunate we didn't, because Oregon DEQ called us and said, oh, Scott, you know what? We made a mistake. Instead of sending you temperature data for the Mackenzie River, we accidentally sent you temperature data for the Deschutes River in Eastern Oregon. So how good that we didn't match that data. So sometimes it's okay not to match field data. When we used the correct data, it fell in line with the other temperature errors. So another quick point has to do with, in terms of calibration, is don't ignore hydrodynamics. This is the Coeur d'Alene Lake, Spokane River, Long Lake going into the Columbia or Lake Roosevelt. We used field data from meteorological data from this red dot, which is the Spokane Airport, and we applied it to Long Lake, which is this reservoir that's in an incised system, which is quite clearly not experiencing the same wind as the Spokane Airport. So this is the model of the Spokane River in Washington and Long Lake or Spokane Lake. And one of the issues was the model matched temperature data. This is in 1991, going from March on the top all the way through October at one location. The data versus model looks good, so you might think that the MET data seems reasonable. Also, for conductivity, we were able to capture the deep interflow of high conductivity water in the summer through the lake, which is from the high groundwater that comes in from the Spokane River in the summertime. So one might think that the model using MET data from Spokane Airport is reasonable. But one of the things that we did, this is the sampling stations on Spokane Lake, is we looked at the wind direction, because fishermen would tell us that it's always very windy along this axis of the lake, but if you want to go water skiing, you go on this axis of the lake. So we adjusted the wind direction in the model. And before we did that, we looked at the dissolved oxygen, and this is model versus data for dissolved oxygen at several different sampling stations at 2 different times. One's August 8th, one's August 29th. And the blue line is the model and the red triangles are field data. And if you look at model versus data comparison, just looking at it, you know, it looks close, but something isn't right. something isn't right. And oxygen is the great integrator of water quality. And one thing we did is we changed the wind direction. And that's the only thing we did. We didn't change kinetics. We didn't change algal settling rates. We didn't change nitrification rates, et cetera, or sediment rates. And by just changing the wind direction at the sampling station LL5, we got this model data comparison, LL4 this, and now it's actually reproducing well the vertical structure of the dissolved oxygen, which is quite complicated. Also going toward the dam, the model and data now tend to be in great agreement, and at the dam, quite good. It's not perfect on the surface, but at those 2 times, what a difference just changing the wind direction made in terms of matching field data. So the lesson there is it's not all kinetics, sometimes it's hydrodynamics. So going on here, a couple comments. How good should your temperature model be? This is looking at Pine Flat Reservoir in California. This is in 1 year. This is at the dam. You're looking at model versus data. The data are the little squares, the model is the continuous line. And it captures a little double thermoclines in July. And this is 1 year, which is very different from the next year. This is also Pine Flat Reservoir, but at a much higher water level year, you notice how the model actually is able to capture the dynamics well, even though the 2 different years are quite different. And if you look at the small numbers show the AME, absolute mean error, and the root mean square error. And then we have Richard B. Russell Reservoir, which is a pump storage project in North Carolina. And Paintsville Reservoir, again, your model should do this well. And then this is Brownlee Reservoir on the snake system between Idaho and Oregon. And this is at 1 snapshot in time, looking at about almost 20 profiles during 1 sampling trip. And there were 1,277 profile comparisons from the time period of comparison. And the model was able to get within 0.7 degrees C absolute mean error and a mean error close to 0. And so if you look at, in general, at statistics of reservoirs, absolute mean errors for all these systems, as 70 different reservoirs, have all been well less than 1 degree C. So you can imagine that I'm going to recommend that if you're looking at how good your model should be for temperature, it should be less than 1 degree C for error. Let's look at another system, Chester Morse Reservoir. Emery Cerovich worked on this system, and just looking at temperature calibration for 1 year. The field data are the dots and the model is the blue. And again, you have mean absolute error of about 0.5 degree C and a mean error of 0. That's what you should expect from a model if your boundary conditions are good. And then this is the typical temperature of the lake at that 1 location as predicted by the model during that time period, as well as the development of stratification and the destratification and lake overturn. Similarly, for dissolved oxygen in Chester Morse Lake, you might notice the field data are very complicated with a spike just below the thermocline due to not only algal growth just below the thermocline, but also strong stratification that occurs at those points. And the model is able to capture that vertical structure reasonably well with dissolved oxygen, model versus data of about 0.5 milligram per liter. And this is the dissolved oxygen over time and depth, again showing the buildup of and changes in DO over time in the lake, which is for most of you from a limnological perspective is probably how you like to see these data. So to conclude this presentation, the proposed calibration targets, how good should your model be? I didn't really talk about water level, but the model should predict water level within about 10 centimeters. Your temperature should be within 1 degree C. Your dissolved oxygen within 1 milligram per liter. Nutrient and algae concentrations, which we didn't get into, should be between 10 and 25% of the range of the field data. And your mean error should reflect no bias. So these are some of the goals that we would propose. And then the final comment is, most importantly, if your model isn't good enough, go sailing, because if you're on a lake or reservoir, you really understand the wind field. And in lakes and reservoirs, it's all about the wind. So I call this the Steve Hood corollary because he's a proponent of that in Lake Whatcom in Bellingham. So I'll stop right there and take any questions.
Excellent. Thank you so much, Scott. Do we have questions for Scott? I guess, oh, Dan's got one. You've got a bunch of questions.
Okay, no, great talk. I think I've seen an iteration of this earlier, but it's always good to see and get reminded about model aspects. I guess my question has to do with that last point about your calibration targets. And oftentimes, in the regulatory system, you'll have no variation beyond 0.2 or 0.3 degrees C. And so I wonder if you could comment about how you might need to adjust your calibration targets or how you deal with that when the sensitivity of the regulatory standards are quite a bit smaller than the calibration kind of targets that you were just stating.
Yeah, I think you're talking about the fact if you use the model in a differential run, comparing two different simulations,
exactly.
You're trying to compare the delta.
Yes.
So that's different than the absolute. How good your calibration is. So I think they're just they're just different.
Yeah. Okay.
And what you're looking at is for trends and indications in the model. But you're right. If you're trying to prove that run A or run B is within 1/10 of a milligram per liter or 0.2 of a milligram per liter, that's pretty fine thread, pretty tight.
Or if you're trying to meet an 18 degrees C standard or something like that. But
yeah, now I think if the model seems to match those things well, I think the question is what's your alternative?
Yeah.
If you have a reasonable model.
Okay, great, thanks.
Thanks, Dan.
Amalia, you're next.
Hi, Scott. Thanks for this talk. It was some really good reminders about what's important to keep in mind with modeling. With the work that I'm doing, we're interested in looking at harmful algal blooms on slightly longer time horizons, like over the course of a summer or even over the course of a year. And to your final point, and looking at these on lakes, and to your final point about wind being a really important factor, I wonder if you have any advice or recommendations for how to think about incorporating wind into models when they're not necessarily, when the time step isn't instantaneous, for example, or on an hour time scale, but how do you think about wind on like a seasonal or an annual time scale?
That's a good question, because usually we're using our model wind data is at a minimum every hour. So we're using hourly wind data or even finer wind data if it is available. I think the big problem with wind data is that if you measure wind at one location, it may not be the same as another location. And so there's great variability in wind, even along one lake. So that variability itself is kind of painful. So I've got a lot of good studies showing where if you're taking wind at the bank over here versus having a wind station out on the lake versus a wind station at the airport, they can be totally different. And so that's just the way life is. You do the best you can. And I think what that implies is that every time you go out sampling, so when Desiree's out there in her kayak in the lagoon on the Willamette, carry a wind gauge and measure the wind at the moment you're doing sampling so that you can always correlate it to a long-term record somewhere else. So, because often we don't have wind data where we're doing our sampling.
Thank you.
Theo, and then Matt, and then we're going to move on to our last speaker.
Yeah, Scott, what's the origin of the cyanobacterial physiology that's used in these models?
The origin?
Well, is it microcystis or is it Anabina dolichospermum, or because they're quite different, and so what sort of growth data and responses to various conditions are used, and where's
That's a good question. So basically, in the model that we use, that the C = W2 model, if you define an algal group, you have about 24 numbers that define its stoichiometry, define its settling rate or floating rate, define its half saturation constants for nutrients, that define its growth rates, respiration rates, temperature preferences, etc. So basically you as a model user formulate your own group, hopefully based on literature values for different groups.
Mm-hmm.
So what
But
we
in
usually
your modeling, have you differentiated a couple of different types of?
I didn't catch that, Theo. You want to repeat that?
In your modeling, have you differentiated between a couple of different types of cyanobacteria, say Microcystis and Anabina or Dolichospermum?
We have not had an opportunity to break that up. I think the most algal groups we've modeled is about 7 or I think we've gone up to 8 different algal groups managing. But keep in mind that if you have 8 different algal groups, different species, you're dealing with hundreds of kinetic coefficients and information that you think you have to know about them. So in general, when we start off a modeling study, we usually start with diatoms, greens, and cyanobacteria as the 3 major groups, and try to include the temperature preferences for a large mixed population of each group. But you could easily break it down into multiple cyanobacteria groups.
Thanks.
Yeah, thanks, Theo.
All right, Matt, and then we'll move on.
Yeah, thanks for the talk, Scott. I really enjoyed it. And thanks for taking my question, Desiree. I was really curious. You said you emphasized the importance of wind on that upper surface. And I'm curious if you have an idea of the sensitivity of the model output to the bathymetric data. So I know it's laterally averaged. It's a 2D models, and I'm wondering if maybe there's less of an emphasis on that lower boundary, and it does less to impact the hydrodynamics or not. Thinking about systems that maybe have outdated bathymetry or so on, perhaps that's important.
Yeah, Matthew, great question. Really, how well your model performs is very much dependent on your bathymetry and your meteorological forcing and inflows and outflows. So it's kind of like if the glove fits your hand perfectly, it's going to perform better. Same thing with the model. If you have good bathymetry, it will perform better. It will do a better job. So the model is very sensitive to bathymetry. Yeah.
It is. Okay. Thank you.
All right. Thanks so much, Scott. Great discussion. Really appreciate it. Our last, but not least speaker, Edgar Rudberg with CD3. We'll be talking about some neat innovative stuff with in response to COVID. Thanks, Edgar.
Yeah, absolutely. And thanks for having me. I want to make sure that everyone can hear me okay. Hopefully my earbuds are working correctly.
Yep, sounds good.
Wonderful. So I'm going to talk a little bit about a program that we piloted in partnership with Ramsey County here in Minnesota, looking at alternatives to in-person inspections to help reduce the spread of aquatic invasive species. So first I'll start with the problem. I'm sure most are familiar with aquatic invasive species as a problem, but then discuss how that problem interacts with COVID, talk a little bit about behavioral science because that's my background, talk about site design and standards, and I'll wrap everything up with our check-in and check-out web hub pilot and some of the results we had from there. So the problem, out west, I know everyone's very concerned about quagga and zebra mussels, but that is only one of the many aquatic invasive species that we are dealing with here nationwide, whether it's Eurasian milfoil, water chestnut, etc. The cost to the economy for that invasive species, or the cost of invasive species to the U.S. economy is in excess of $146 billion per year. And what we're finding that is existing tools that we have in the toolbox to help stop the spread of aquatic invasive species are costly and cannot scale. Obviously, in the West, we do a very good job at having interstate checkpoints when you're going from one state to another to get decontaminated. However, we're less good at stopping the movement of aquatic invasive vegetation, for example, within the state. And what we found is that, in particular, boaters need the tools to empower them to do the things that we're asking them to do. When we complicate that with COVID, we're not looking at many of these types of interactions currently, right? What the typical, at least in the Midwest and the Northeast, is we'll have volunteers. They could be from lake associations. They could be hired staff from DNR. Ends up being an educational experience for the boater. But we found, obviously, between shortages of staffing and then just people not wanting to have these types of interactions, was that this just wasn't happening as much. And so we needed to find alternative ways that we could educate boaters and empower them to clean, drain, and dry their boats in the absence of those interpersonal communications. So this is my doctoral dissertation, pretty much summed up in one slide, which is, in behavioral science, we have educators, whether it's Sea Grant, state agencies, nonprofits that are pushing behaviors to, particularly in this case, boaters. And we are asking them to clean, drain, and dry their boats and trailers. And in the state of Minnesota and many other states, we're also asking them to dispose of their bait. So boaters will then plan to act and do that, but we often find when it comes to the actual actions that day boaters fail to take the actions needed to prevent the spread of aquatic invasive species. So we looked to the research that is out there, and we found in the literature that there are many different reasons that are identified. And this was a great research study done by the Canadian Council of Invasive Species. If you all aren't interested, or not interested, but if you all aren't familiar with qualitative research methodologies, what this is, people get interviewed, and the more often times a word comes up, the larger that word ends up showing in the word cloud. So equipment and a station was the number one answer that people gave as to why they don't always clean, drain, and dry. We also looked to the State Organization for Boating Access to understand what their recommendations are for getting boaters and empowering boaters to do what they need to do to clean, drain, and dry, and thereby reduce the spread of aquatic invasive species. So they have a best management plan or best management practices document that really walks site designers in how to create a site that empowers boaters to clean, drain, and dry. So part of that is education. So you'll see that part of this, obviously we have signage going into the water body and then signage coming out. But also you'll notice on the right-hand side here, we have areas where they're demarked to allow people to pull their boat plugs, to put in their boat plugs, or to pull out, where we have another educational signage and area where boaters then can do what they need to do to remove AIS and tie down their boat. So at CD3, about 5 years back, we developed a cleaning station that goes right at the boat launch. Reason that it's supposed to be right there is that we found research in Michigan showed that if it's a cleaning station is across the street even, you have a steep decline in use. For example, a pilot in Michigan showed that a cleaning station across the street only had 4 uses within 2 years, and they thought of those uses, 2 of them were to clean their car. So it needs to be extremely convenient. It needs to be where people tie down. And in Minnesota, for example, we're very passive aggressive. And so we'll look at you sideways and we'll tap our foot and be stern. If you don't have kind of an area that's demarked to allow you to do those things. And that's what's shown here in the upper right-hand corner. And it's also important to include things like waste bins. recycle bins. So yet one more reason for people to stop and use a cleaning station, but also to leverage language and images that are vibrant and really focused on the behaviors that we're trying to catalyze, which is cleaning, draining, and drying. So what we found that when you use the State Organization for Boating Access, that manual to design a site, you include signage and you include cleaning stations. This is showing a pre and post test. So on the right, these two gray columns show that we had about a 20%, somewhere between a 20 and 21% violation rate at sites without the extra signage and the cleaning stations. These two on the left are experimentals. The blue, actually the 16.5% violation rate, that was after the first year. And we found that after the second year, their violation rate drops down to 6.3%. This site here, 20.3 was the first year where there wasn't signage or cleaning stations. And then the second year we dropped to 9.9%. So we found that we can, just with signage and a cleaning station, we can reduce violation rates by over 70%. However, there's obviously still that 6 to 10% that we have violation rates that we still need to find ways to use Jedi mind tricks, if nothing else, to get people to do the things that we're asking them to do. And that's where community-based social marketing comes in. The idea with community-based social marketing, if you're not familiar with it, is we overcome behavioral barriers. Now we're trying to overcome behavioral barriers with the types of stations we've created to enable people to physically be able to clean their boats. But we also want to use other tools like a social contract. A social contract is a statement to the public that you will do something not only now, but in the future. And that's enabled with things like social media. And ultimately, our goal is to decrease the spread of aquatic invasive species, increase compliance with AIS regulations, and increase the use of available tools. So what we created then was a handheld system on any smartphone. We considered a screen kiosk, but we realized that there's the possibility of vandalism, of damage, of other things, and they're incredibly costly. And more we're finding that everyone has a screen in their hand, right? So, and we're also finding that particularly with COVID, people are more comfortable with using QR codes for menus in restaurants, for many different things that we used to grab things and touch things that we're not doing anymore. So we created this check-in and check-out web hub that's specifically based on community-based social marketing. So what we're doing is we're connecting then our educational platform that you can go in and you can check it out on your phone, which is ramseyais.com, and you select the lake that you're at, whether or not you're checking in or checking out, and then you select the type of boat that you have. And then there's a series of instructions based upon whether or not there's a cleaning system or tools to help you clean your boat. And then it gives you the instructions specifically based on whether you have a personal watercraft, a fishing boat, a pontoon, etc. And then we have a social contract at the end. So you say, hey, I'm willing to do this now and also in the future. You sign with your finger. And then we have the ability to push that information out to social media if they so choose. The other thing is we can connect that program with, at least for now, a carrot. So you get an incentive. If you check in and check out at a certain site, you can get the incentive of what we use, which was a coupon at a local sporting goods shop. So the sporting goods shop shows goodwill with the local county, but they also get a coupon that brings customers in. We geolocate that so you can't be sitting on your couch and getting coupon over and over again, and that you have to be in the area and you can only do it a certain amount of times. And then, like I said, we have instructions based on the types of tools that are available at the different sites. For example, you can see the sign here, and then over on the right-hand side is a close-up that shows some of the tools that they have just to help boaters, at least if nothing else, We know in Minnesota and in general in the nation, our average boater is 65 and older. So getting on one's hands and knees and pulling debris, leaves, milfoil off your trailer becomes increasingly difficult, and then a marine brush. Our stations have a wet dry vacuum, tethered tools, again, a grabber tool, brush, and then a boat plug wrench, and then compressed air. So all the tools anyone would need to clean, drain, and dry their boat. And this one happens to be solarized, so it can be entirely off-grid. The other thing is we want to ensure that we're using pictures over words. Sign fatigue is a real thing. When boaters go up to a boat launch and they say 50, in the case of, and not to throw the Minnesota DNR under the bus, but they do brown and white signs, and they're not particularly visually appealing. So if we can use signs like the upper left photo here, that shows a lot of graphically of what we're asking people to do, and keep the message really simple, clean, drain, and put in your plug. Also, we find that people always, not always, but often follow lines. So if you have lines that are directing people to cleaning stations, to where they should stop here, we're finding that 90% of the people actually stop where we're asking them to stop. So then they stop there, they get the visual cue from the cleaning station, which is essentially a educational kiosk, right? It has instructions on it. You can get more information on aquatic invasive species, etc. And again, delineating that area so that people feel comfortable that they can pull off and stop there. And then we're focusing on two different areas, right? There's the kind of the staging area before you go out on your boat, focusing on the kind of the getting ready with a simple calls to action, again, delineating a space and having some equipment there, such as the tools you see here to help people get their boats ready and make sure there's no vegetation on it. And then as they leave, again, this kind of containment strategy, having a cleaning station or a cleaning area within 150 feet of the boat launch, direct with arrows, lines, and use these and leverage them as an educational kiosk. So I think I missed one. Okay, so what we're able to do then, looking to the future, is having unique identifiers. So parks, for example, Ramsey County, they own all their boat launches. And so they can take payments, you can check in, you can put your boat number, zip code, vehicle license number, have the ability then so that we can track people if they're coming in or coming out, and if they have indeed checked in or checked out. And that opens the opportunity for enforcement. Right now, we're wanting to be fun uncle, if you will, rather than big brother. So we're really focused on those positive incentives, but there is the possibility to get more people to follow what is county rules. There is the possibility of violations, and the ability to use this as enforcement. Right now, it's being entirely used for educational purposes. So in the end, not only are we empowering the public with cleaning systems and tools, but also trying to get an educational platform that overcomes these barriers to action and allows people to be social and state that to the public and to their friends that they will clean, drain, and dry their boats. And with that, hopefully I kept it under 15 minutes because I know that was the goal, and I will open it up for questions.
Excellent. Thank you so much, Edgar. Matt, looks like you still have your hand up. Do you have a new question? No. Okay, great. Edgar, I have one quick question while folks are generating theirs. I'm just curious, how much does one of these stations cost at a site?
Sure. So, we have a range of stations. The very small one that's not internet connected is about $1,500. versus the solar unit here that obviously has vacuum and compressor. This shipped to Oregon is probably around $31,000 installed.
Okay, thanks. Have you seen any analyses either within Minnesota or otherwise where folks have sort of quantified the economic impacts of invasive species on lakes? Or, I mean, I'm just curious, sort of, yeah.
Yeah, so St. Cloud State University in Minnesota looked particularly at home values on lakes, particularly with different infestations. And what they're finding is a direct correlation between the value of one's home and an infestation of an invasive species. We're particularly concerned in Minnesota about starry stonewort. That creates, dense vegetation mats in different types of lakes and really impacts boating as well as angling. And you're seeing just the value of those homes really, really fall. We also know that invasive species, particularly, we have a large lake in central Minnesota called Mille Lacs. And the introduction of zebra mussels and spiny water flea, they're consuming 75% of kind of the food chain, if you will. And that caused the walleye fishery to crash. And the economic fallout from that in just one county, of which there are 3 counties around the state of, or around that lake, one county lost $20 million in a tax base because of the walleye closure. And that's just for one summer. So they had resorts closing. It just totally changed the recreation on that lake. And so it's obviously, the context is specific to the lake and the type of invasive species, but large negative economic impacts.
Okay, excellent. Thank you. Amalia?
Thanks for this really interesting talk. I wonder, in your presentation, you shared a figure that showed the decrease in, I think it was violations after implementation of some kind of intervention to sort of nudge people to adhere to this clean, drain, dry protocol. And now that you've kind of gone through the different options for it, I'm wondering if you can say a little bit more about what exactly that intervention was, and are you finding like how you stack these interventions, everything from like a sign to a whole station? Are you finding very big additive or synergistic effects from these?
Yeah, absolutely. So what we're finding is what we tried, and how we found this, if you all aren't qualitative researchers, we had secret shoppers essentially. So people sitting at a picnic table, I'm having lunch or I'm reading a book or something, but they're really spying on you, as creepy as that is. But what we found is when you redesign a boat launch, and that's what that was, is following those best management practices from the State Organization of Boating Access, is, with the signage and putting the sites, kind of those areas, those demarked areas where you can have gotten the emotional space, if you will, to clean, drain, and dry your boat. We're seeing a drop. And then we're seeing with the added, the tools such as the CV3 station, we're seeing that extra precipitous drop in those violation rates. So it absolutely is tiered as you invest further in your site. And for example, Hennepin County, which is a western suburb of the Minneapolis-St. Paul area, they even have almost the lighted signs like you would see on a highway. So, I mean, big signs, a lot of stuff. It's not for every site because it's a little bit over the top and you wouldn't want it in a more rustic area, but they're seeing a huge decrease in those violation rates when they really are investing in those sites.
Thanks.
Any other questions for Edgar? Theo, you're next.
Edgar, yeah, good. Really interesting to have the behavioural aspect. So one of my bugbears is when I go up the trails, dog people take their bags, but then they park them on the side of the trail, promising themselves they'll come back and pick it up, but somehow they often don't. And so I'm wondering, if there's somebody who has a boat and really only takes it to one lake, maybe they would argue to themselves that the cleaning and stuff isn't that important, but in practice they probably do go to other lakes at times. And is that a behavioural problem that undermines some actions?
Yeah, I mean, I love the garbage or the refuse example. Disney's done a great job of identifying exactly how far between garbage cans you need to put it so the refuse doesn't get on the floor, right? It's 10 feet or 15 feet. I mean, it's crazy. But they've done that research. That's the same thing here. I mean, if you think you're not the problem, you're not going to do a solution, right? And so what we try to do is not always have the conservation pitch, right? Because if we talk about conservation and things, we're hitting the same people on the choir. It's that 30% that's going to do the right thing for the environment every time. What we often will do on these is put a big sign that says free boat cleaning station, right? And so we try to tap into the idea of, hey, I've got this $70,000 bass boat. And this is particularly in Texas, right? You got a $70,000 bass boat that you want to keep sparkling and clean. And so we try to target different value sets that aren't the conservation value set over and over again. So even if you're going to only one lake over and over, you still don't want your sparkling nice boat to be dirty or smell in your garage. So I would hit those folks with a little bit of a different message so that they can, have this there. They can clean up their boat right then and there. And whether or not they think they're doing the right thing for conservation, I'm a little bit Machiavellian about it because we're getting to the goal that we want regardless of the driver for their behaviors.
Good plan. Yeah, thanks.
All right. Well, we are at the end of our speakers. We have a few minutes left if folks have other questions or discussion before 5, or feel free to peel off as it makes sense for your schedule. Thanks so much to all of our speakers and to all of you for participating. Really appreciate it.
Thanks, Desiree.
Hey, Desiree, I was going to ask a question. You mentioned that there's that cold temperature plug at the bottom of the lake, and you also mentioned that it's high in nitrogen. How did you deduce that?
Oh, I didn't discuss any of the field data that I collected. I collected in 2020, I was collecting nitrogen and phosphorus samples from multiple locations in the lagoon and at different depths. So it had at the surface, at the metalimnion, and then at the hypolimnion. And so the hypolimnion consistently had higher nitrogen than our control site upstream and anywhere else in the lagoon. So that and it didn't have elevated phosphorus. So it's a hypothesis. It's not, confirmed, but it definitely DO was really low. So it could be that DO is low and it's releasing, sort of kickstarting the nitrogen cycle and releasing that nitrogen, but it wasn't doing that for phosphorus. So the two explanations are, one, it's coming in with groundwater, or that the soils have nitrogen but not phosphorus in them.
And was the river, you were thinking it was river sourced because of the temperature, and would that be bringing in nitrogen?
Well, so
at level?
I've been trying to track that with the USGS gauge data, but it just doesn't, the data don't, I haven't been able to really get a sense that the nitrogen concentrations we're seeing in the winter line up with the concentrations we're seeing in the bottom of the lagoon. But there's so much happening between, January, February in the river and August in the bottom of the lagoon. It's hard to say whether or not that's meaningful or not, the fact that they don't really line up.
Okay.
Yeah, it's a great question. Yeah. There's so many mysteries. That place is full of mysteries.
Desiree, just to follow up, I guess, on that, don't want to beat this too much, I guess, but I think I mentioned this before. So you mentioned regional groundwater is at about 12 or 13 degrees Celsius, but are there pockets maybe that are even colder? I'm thinking, because that's just because I live in the area, that's kind of near Crystal Springs. I know that's exceedingly cold, and I'm not really sure what the temperature is coming out there, but it may not be, could be colder than regional groundwater, or it could be, I don't know.
Yeah, I'm not a groundwater person, Dan, but I've asked several people now who are groundwater people, and they're like, there's no way to hypercool groundwater. It's all based on the long-term average of the air temperature, which is 12 to 3 C. And they're like, there's no way to get it colder than that. I'm not saying that's not true. That's just what I've been told. So we looked into doing groundwater study, and it was going to cost $500,000. So we are working on, plan F at this point to try to figure out. I mean, the ultimate question is, I actually don't really care where the water comes from. The question is, if you start bringing it to the surface and pumping it, will you exhaust that cold water supply? Yeah. Will it refresh every year? Or is this like long-term groundwater that's been parked in there that we're going to lose? So it's more of a, I mean, it's maybe cynical of me to say I don't really care where it comes from. But everything I've been told anyway is that it can't be colder than what the long-term average is. But I don't know.
No, thanks.
I'm not a groundwater person. All right. Well, thanks everyone. I think we're done here. And yeah, maybe see some of you in a couple weeks. Have a good Thanksgiving and a good evening.